Okay, so it's Monday. This is the second week of classes, and we're going to be picking up with the second half of, or really the last part of Chapter 2. This last material will be the last material that will be part of your lecture exam. Now, that lecture exam, if you look on your syllabus, that lecture exam will be not this week, but next week. But next Monday, we don't get together. Right, next Monday is Memorial Day, and so I'll be seeing you all on Wednesday for lecture, and then not again until the following Wednesday, at which time we'll have our first lecture exam. That exam will be a full-length exam. You'll need an 884-E Scantron form. That's a double-sided green Scantron form, and a pencil and an eraser. And that's all you'll need, and that's all we'll do for that time together is you'll come in, answer any questions we have, and then we'll take the exam. And then after that, you're free to go.
You said double-sided orange, right?
Nope, it's green. 884-E Scantron. The orange is a par score sheet. They gave you the wrong one. So go back and tell them you need the Scantron 884-E double-sided green form.
Okay, those are the ones I
Yep.
thought it was the other way around.
Nope. And on vocabulary on that exam will be what? 1 through 20. I'm going to go through 11 through 15 with you today, and then the last 5 slides on Wednesday. Content, Chapter 1, which was everything up through the PET scans that we did the other day. Chapter 2, the chemistry, including what I finish up with that today, and then also what we did in Lab 1, directional terms, body cavities, all the things you're being quizzed on this week in lab is also a very important part of that exam. Now, if I look at this packet, I just gave you the student worksheets. Everyone, you purchased this with your package, and it wasn't yet available. And what you have here are some beautiful review sheets, some worksheets to work through this material. Now, remember that this textbook is being used for both Biology 105 and for Biology 106. And so there will be content that we will not necessarily cover this semester, but that instead will be covered next semester. So your rule of thumb is this. When doing problems on mastering, when doing let's say we're labeling the muscles later on in the course, and you get to an image where you're being asked to label 35 muscles, and 25 of the muscles we've talked about and 10 of them we haven't talked about, guess what? You're only responsible for the 25 that we have discussed. So I know it's a little bit of confusion for you. So as you're looking through these worksheets, if you're looking in Chapter 1 or in Chapter 2, and you see a concept that we have not discussed, you are not responsible for it. Okay? And so that's what you want to do. You want to let the PowerPoints and let my presentations, let my PowerPoint notes be your guide as to what things to deal with and what things not to deal with. And as we go through the book, it'll become more and more clear to you, I think, the things that you don't have to worry about. So this is your work product. This is not something you're going to turn in. This is simply another way for you to be successful in the course. My recommendation would be to 3-hole punch this as you need it. So keep it safe. And then right now we're doing chapters 1 and 2. Throw it through a 3-hole punch, stick it with your PowerPoint notes, stick it with your lecture things, and then when we get to exam 2 and you've got lectures 3 and 4, or chapters 3 and 4, grab those materials. So just keep it in a nice, safe place. This would have normally been in a bound notebook as a part of your bundle. It wasn't yet published. They've just sent them to me overnight shipping. So now you have these, okay, to take advantage of. So take a look at these. No points for doing them. They were free additive to your textbook, and so I hope that you will enjoy these. I will be looking forward to hearing back, getting feedback from you about those as well as we go through the semester. So here we are. This is the second half of Chapter 2. Right now, let me entertain. Are there any questions related to Chapter 1? Anything related to the first half of Chapter 2? Over Chapter 2, we've talked about what atoms and molecules and macromolecules are. We're about to discuss the macromolecules in greater detail. We discussed pH a little bit, buffers just a touch. Anything in that conversation at all that needs to be clarified or restated for you? Anything at all? Feeling pretty comfortable? Okay, in the next half hour, we'll have all the information for the first exam. And then you'll have the next 10 days, really 9 days, to really pull it all together. Has anyone had trouble getting on to mastering? Is everyone on mastering? I think I've got 39 students on mastering. I think I'm looking for one more person. Can't quite figure who it is. So mastering is going well. In there, you saw a homework assignment that was due last Friday. It's just kind of an introduction to mastering. I think I saw most of you did that. And now you're going to see a homework for chapters 1 and 2. Those 2 homeworks, you can do them over and over and over. They're there to help you master the material for exam 1. There was also the My Readiness test. If you didn't do that yet, I had a deadline of tonight to get that done. That was that pre-test. That was a diagnostic test. You're going to get 10 points for doing that test. Okay? So go in, do the very best you can. It's timed at 1 hour, 70 questions. The score you get is not the score you're going to receive for a grade. You will get all 10 out of 10 points for doing the quiz to the best of your abilities, okay? But go in and make sure you did get that done before midnight tonight if you have not already done that. Most students, I think, have already done that as well. Okay? Anything else you can think of? Logistically, lab, lecture, anything at all? We're kind of getting to the routine, right? You've now had a week under your belt as far as the classes. You know your routine. You know your classes. You know where your study breaks are. You know about room 249 upstairs. If you were thinking about getting a tutor, I hope you've already contacted the tutoring center. I hope you've already identified room 249 upstairs. That's the little study room outside the labs where you can go to review models and things. You probably don't feel the need to go do that yet, but I assure you in about 2 or 3 weeks when we get to the muscles and the bones and the other systems, you're going to want to have into your schedule some time to stop into that room and review the models. So you might as well establish that habit now rather than wait 3 weeks when it's now necessary. So go ahead and just find that room, step in, feel comfortable in there, use it as a great place to study. Okay, let's go back to vocabulary. Start with vocabulary and go back to slides 11, I believe is where I need to pick up with you. Again, remember the vocabulary. The best way I know of learning this is just using flashcards. And by using flashcards, you'll put the term on one side and then the meaning on the other side. You do not need to worry about the example. It's there as something that you can help remember it, but I won't be quizzing you directly on the example. So if you see a cell that ends in blast, fibroblast, we'll be talking about chondroblasts and osteoblasts later in the course. Those are all cells from which something else germinates or buds. So chondroblasts are the cells that make cartilage. Osteoblasts will be the cells that make bone. Fibroblasts, as we'll see, are the cells that will make fibers and connective tissues. Okay, so we'll see this over and over. So blast, germinate, or bud. Brachy, we know the brachial region is the arm. No surprise there. Brady, slow. What's the opposite of Brady when it comes to heart rate? Tachycardia. You'll learn later on. Tachycardia is a rapid heart rate. Bronch, air passages. The bronchioles, the bronchus, the bronchi, these air passages through which air passes. Okay, so vocabulary. There's 2 of our 3. Let's go to the next one. Buc. Okay, you'll hear buccal or buccal. You hear them both pronounced. That's referring to the inside of your cheek. That was one of your directional terms as well. The buccal region, the inside of your cheek. Burse or burso, purse-like structure. I always think of it more like a gel pack, if you will. The bursa are these gel pack regions around your shoulder, around some of your other joint spaces. So these bursa. And I've got it here as a purse, kind of as a gel pack that goes around your joint spaces. If you had bursitis, you would have inflammation of the bursa, of this gel pack area. Calc, if you see calc within something, it's referring to calcium or limestone, really the same thing. So if someone says you have hypocalcemia, we'll see over and over that emia refers to a condition of the blood. So emia, blood, and hypo, low or under, and calcium levels. So you have a low level of calcium in your blood, hypocalcemia. Callus, if you have a callus, you have a thickened area of skin. So callo meaning thick, and finally for this slide, calce, a small cup. We'll see when we get to the kidneys that there are these structures called a calyx or calyces, plural, that are these small collecting cups for urine. Okay, more about that when we get there. 13, capito. Capital punishment, off with their head. Okay, that's the way I think about it. There is a structure on some bones, the head of the bone, called the capitis, toward the head of the bone. If something is carcinogenic, it causes cancer. Carcin referring to cancer. Cardio, the heart. Cardio, the heart. Cardiogram would be a viewing of the heart. All sorts of terms here, right? Cardiac, we already said pertaining to the heart. And carpo, you know your carpal region is the wrist. There are a bunch of bones in your wrist called the carpals. So carp or carpus is referring to the wrist. We've already seen this term. Catabolism. Catabolic reactions were metabolic reactions in which things were broken down. So a breakdown of things. So cat referring to breakdown. We know this one as well. If something is caudal, is going toward the tail. So cod means tail. Cephal, the head. Cephalic, toward or by the head. Centesis, surgical removal or surgical puncture to remove fluid. We've all heard of amniocentesis. It is a surgical puncture into the amniotic sac from which we remove fluid and then test it for issues with the baby. And then cerebro, brain. Okay, so the cerebrum, the cerebral area is your brain. Last one for today. Cerv. We know this one already. The cervical region is the neck. So cerv or cervix, anything like that, referring to the neck. Chondro, we saw this with hypochondriac region, right? Chondro referring to cartilage, and there's a fair amount of cartilage in your ribs, so you'll hear chondro referring to cartilage or gristle. If you're eating next Monday on holiday when you're having some chicken or some meat, and you're kind of chewing along, and all of a sudden you kind of get this chewy section, right? This kind of gristle, that's cartilage. That's the cartilage that's been ground into the meat. Chloro, green. Chlorine, chlorosis, chloroplasts, all those chloro terms that you learned in maybe a plant biology course, referring to green. And then chrono, if someone has a chronic condition, it's going to last for a very long time, right? So chronos, time. So those are the first 15 slides. Again, you want to have your slides made up and go all the way through, I believe it was cusp, through 20. So you want to have through 20 on the first test. And again, I talked to you about how I would test this last time. But for example, back to around slide 3 or 4, I said I might ask you, what does ambialgia mean? And you would tell me for 2 points, pain on both sides. Now I will quiz you I will tell you right now that on my test, if I said to you, what does ambialgia mean? Right? Ambialgia. I would ask you to tell me what ambi means first and then tell me what algae means second, just so I can make sure that you know which one is which. So on the test, even though you would say in everyday language, you would say pain on both sides, I would ask you on the test to put in the first blank, both sides, ambi, and then the second term, algia, meaning pain. Okay, so I will ask you to kind of reverse it just so that I know that you know, with no problem, which term means which. So that's going to be on the test, and I'll tell you about that when we see that. The test will be largely multiple choice, but there will be some fill in the blank, and the fill in the blank will definitely be the vocabulary portion for sure. So I will have some maybe 10 or 12 terms out of those first, how many were there? First 20 slides, about 100 terms, right? 80 to 100 prefixes and suffixes. And of those 80 or 100, I might ask 20. Okay, I'm not asking all of them. I'll ask about 20 of them. And then that'll be part of the vocabulary portion of the exam. The other part that you can expect on the exam would be some pictures, things on PowerPoint, things that we've been looking at together. I may have a picture up on the screen and say, what is that? That could be fill in the blank. And then there will always be a case study, and you really can't study for this case study. I'll just tell you a story, you'll read through it, and from it, you'll tell me what you learned. So those are the things that you'll see on the first test. Don't forget also that there will be that Darley article, that very first page as you open up into your supplement. There was that one page on study tips on how to get ready for a biology course, and definitely if you haven't done that, read through that just 2 pages, read through the article, and ask yourself, what are some things you might do differently this semester, having read that article, that will help you. Okay. So as I still learn this new program, who's had a chance to go to YouTube and watch any of these? Have you found them to be of good quality, useful to listen back to the lectures? I hope so. Again, this is new technology for me. I'm kind of getting my groove down here and figuring this out. But I'll get a little bit fancier as we go through. But I hope you're finding this to be a very useful way of reviewing the material. Okay, now let's go over to back to chapter 2. And we had gone through atoms and isotopes and electrons and protons and neutrons. We discussed hydrogen bonds, ionic bonds, covalent bonds. Covalent bonds come in 2 types, right? Polar and nonpolar. We talked a bit about enzymes, pH, salts, buffers, and water. And that brings us now to this place where we pick up on inorganic compounds. I just flipped through that in 10 seconds. Anything in that content at all? Protons, neutrons, electrons, isotopes, chemical bonds, ionic or covalent. Anything in that conversation that needs to be restated or explained?
Are we going to need to know for like sodium and how many protons?
The question is, do you need to know that sodium is the 11th element and chlorine's the 17th? I'll always tell you. You'll get tired of it. Between the pre-lab and the lab, you're going to have done it. But no, I will always tell you that carbon is number 6, oxygen is number 8, or something like that. So you don't have to memorize those numbers at all. Anything else related to the chemistry section so far? Okay. Well, we're at that place of atom to atom where we're talking about the different types of macromolecules. Now, how many different types of macromolecules are there? The macromolecules. 4. Name them. Carbohydrates, proteins, lipids, and nucleic acids. And that's exactly where we're going to walk over these next few slides. We're going to discuss what are these different molecules, what are some of their properties, and then we'll continue to have this information as we move forward into chapter 3. So another word for these 4 groups of macromolecules would be organic compounds. Some of you in the future may take organic chemistry. And organic chemistry will be the very study of carbs, proteins, lipids, and nucleic acids, right? What is their biochemistry? What is organic chemistry? Most of you will start off chemistry in an inorganic chemistry class. And you won't spend a lot of time in an inorganic class talking about lipids and proteins. You'll instead spend your time talking about inorganic compounds, for the most part, things that don't have carbon. So you'll spend your time talking about iron and magnesium and chlorine. So by definition, inorganic molecules are going to be smaller molecules, and they're going to be molecules that do not have carbon. That's their big thing, right? They lack carbon. Now, the only exception to the rule that I'm aware of is carbon dioxide. Carbon dioxide, it's a very small molecule, and therefore it is considered inorganic. So even though it is containing carbon, it is still considered inorganic. That's the only exception I'm aware of. So here we go. These organic compounds, they all have carbon in them. They are a part of our biochemistry, right? Our organic chemistry. And we already know the 4 classes. We've already said them today. So we have carbohydrates, lipids, proteins, and nucleic acids. I'll go through these 4 groups one by one, give you some examples of each, understand what each is important in our body, why we need them, and that'll be the end of the material for chapter 2. Let's start off with carbs. Carbohydrates, we hear about them all the time. They're in the news. They're on our food labels. What are they? Well, different examples of carbs would include sugars. So name some sugars for me.
Glucose.
Yeah, I heard glucose. When you're looking at your food labels, you may not see glucose. You might instead see sucrose or dextrose. And the big thing is that they end in ose. So most sugars are going to end in OSE. Okay, so if you're looking in your food label, you see an OSE on there, you're pretty sure it's a sugar. Dextrose, sucrose, fructose, you name it. Then there are starches. Where do we get starches from? Give me some example of starches in your diet.
Potato.
Yeah, potatoes. Yeah, corn, right? The starchy vegetables. I heard bread, right? Rice. Those are all starches. And when you consume a baked potato, your body very quickly breaks down that baked potato into basically sugar, right? You might as well just be eating pure sugar. Your body really doesn't see it much differently than that. And then there's glycogen. This is the molecule we don't talk about quite as much perhaps, but this is the way by which your body stores sugar. So your body, one way it's going to store energy is to make glycogen. So as you consume sugar, as you consume your daily diet, your liver and your muscles are going to take some of that extra sugar and store it as a long chain of sugar called glycogen. I'll show you a picture of it in a minute. But glycogen is a storage molecule, and maybe you haven't had anything to eat since lunchtime. So your body right now is in need of some more sugar. So right now your body is breaking down that glycogen that's being stored and is releasing sugar into your brain, into your bloodstream. So your brain is fed and you're able to concentrate. Now, the carbohydrates are the primary energy we use for life. So when you look at a food label, you will see on it calories, right? And calories in other countries, they don't put calories there, they put the word energy, right? So you go to Europe, and on the can next to the food label, it doesn't say calories, it says instead energy, and then next to energy, it'll tell you the number of calories or kilocalories in that food substance. So it is the primary molecule that gives us energy. Now we're going to break carbohydrates up into three basic groups. This is a very basic understanding of carbs, but we're going to have monosaccharides. That is one unit, mono. So these are single sugar groups. And then there will be disaccharides, and I'll give you examples of those. Two sugar groups linked together. And then finally polysaccharides with three or more. So one group, two groups, or three or more groups all linked together, that would be a polysaccharide. So let's start off with monosaccharides. You're not going to be responsible for memorizing or recreating any of these images. So right off the bat, you don't have to be able to recreate this monosaccharide. But I want you to see that by definition, a monosaccharide is a molecule that has between 3 and 7 carbons. Okay, between 3 and 7 carbon atoms. And those who've had a little bit of chemistry would recognize this easier. If you haven't, it's okay. But on this molecule, every one of these little corners, every one of these little angles is another carbon that's not shown, that's understood. So there's a carbon there for each one of those. So this would be a 6-carbon molecule, right? Each of these little bands is another carbon, another C that's not there. Now, examples of monosaccharides that you've heard of, glucose and fructose. Glucose, the name you'll see more often for glucose on a food label is dextrose. So if you're looking at a food label, it may not say glucose, but it may say dextrose, and you're thinking, okay, what is that? It's the same thing. Okay, and then fructose, a common fruit sugar. Common sugar. You'll see high fructose corn syrup, right? A lot of those names are definitely on your food labels. Those are monosaccharides. Monosaccharides are taken into your body and they're used very, very quickly. The body doesn't have to do much with them. They're already in their single form. So the body can take them in and use them very, very quickly. Disaccharides. Two of these monosaccharides linked together through a chemical bond. So one example is sucrose. So sucrose, this is your common table sugar. Okay? Good old table sugar. The 5-pound bag of sugar you buy at the store, that is sucrose. What is it chemically? It is a combination of glucose holding hands with fructose. So two monosaccharides linked together chemically to make sucrose. And here you can see the chemical bond. Okay, so we've got one glucose linked to one fructose. Again, you don't have to recognize these units, but you do notice that they are linked together and that each of those little lines in here represents a chemical bond. So they're electrons that were rearranged and molecules that are sharing or are taking over electrons in order to rearrange themselves. So no sucrose as an example of a disaccharide. And then we get to the polysaccharides. Many, 3 or more, oftentimes hundreds, thousands of these molecules linked end to end. In the bottom, this is a cartoon of glycogen. Again, you don't have to memorize it, but you see there's a glucose unit, a glucose unit, a glucose unit, a glucose unit. And they're just holding hands. They're holding chemical hands as a long chain. And you see it has many, many branches, so overall, it just has this really funky arrangement with all these branches of sugar molecules. This is very, very similar to what starches look like as well. So you hear about complex starches, right? Complex starches are molecules that you can eat now, but it takes the body longer to break them down. So what does a marathon runner do? The night before, they carbo load, but they don't eat simple sugars. They're going to eat complex sugars, and the idea is that the body will take those complex sugars in. It'll take longer for them to start breaking them down, and then the next day, while doing their exercise, they'll have more energy available to them during the long haul. Okay, but if they're not going to be eating baked potatoes, and they're not going to be eating sugary things the night before, that's going to cause the blood sugar to go up very, very quickly and then to crash. But complex carbs, that energy can be longer sustained. So again, these are the major molecules for storing energy. So I mentioned glycogen earlier, right? This is glycogen on the bottom, or starches. These are long chains. So we consume foods that are polysaccharides, and our body makes polysaccharides. Our body makes this glycogen to hold on to energy and to make it available when needed. Now, how are these sugars made? How do we combine two simple sugars, two monosaccharides, into a disaccharide? Or how do we make a long chain to make a polysaccharide? Well, the basic idea here, condensation and hydrolysis. Before we even describe the chemistry of this, I want you to ask yourself, what do those words possibly mean? Or what can I make sense of this? So, condensation. In your everyday experience, what does condensation mean? Say it again. When water sweat, yeah, okay. What else was someone going to say? Evaporation. Okay, I'm with you. You're picturing what? Condensation on a glass, right? You just poured a soda or something and you notice the water's condensing on the outside. When else in the kitchen, perhaps, do we have condensation going on? I'm picturing, yeah, on the stove, I'm throwing my pasta in the big pot of water, put the top on it, and I lift up the lid, and what do I notice? Water has condensed. So my little word picture, my simple little mind has to have a picture. So my picture says condensation means that water molecules are coming together, right? Water is coming together to form these beads of water on the inside of the pot or on the outside of my glass. Right? Water's condensing. Hydrolysis, hydrolysis. Hydro, water, lysis, to break. So hydrolysis is the breaking of water. So now we've got these two words, right? Condensation, water coming together, and hydrolysis or hydrolysis, where the water is being broken apart. Now let's figure out what this has to do with this. In condensation, chemical reactions, two compounds are going to be joined together. Two compounds will be joined together. It's like two monosaccharides being joined together to make a disaccharide. And oh, by the way, when that chemical bond is formed, water is formed. Water is a byproduct of this reaction. So water is being created. Okay? Again, condensation, water is being put together or being made or created here. Now, in a hydrolysis reaction, it's the other way around. Two compounds are being broken apart chemically, and as a result, we're going to see that water has to be broken apart for this to happen. So hang in there. Let me show you this in a more of a picture. So this is a condensation reaction. Again, you'll never have to recreate this chemistry, but you need to understand the basic ideas of what's occurring here. So we have a monosaccharide, a glucose, and what it doesn't have here is plus, so plus another glucose. So glucose plus glucose, and I see the chemical reaction, I see the arrow. Now remember, an arrow tells me that a chemical reaction has occurred, and we know that when chemical reactions occur, electrons are rearranged, molecules are rearranged, and in this reaction, we form maltose. Maltose is another disaccharide. Maltose is glucose plus glucose. A moment ago, what did we have? Sucrose was glucose plus fructose, and now I'm telling you a second disaccharide that you need to keep track of is maltose, which are two glucose molecules linked chemically together. Now, when these two glucose molecules combine, I want you to see they've highlighted it here for you in blue. There's an OH group from one glucose and a hydrogen from another glucose, which are going to be removed to form water. So I have created my disaccharide, I have linked them together chemically, and in doing so, I've also created water. This is a condensation reaction. Okay? Now the other direction is hydrolysis. So if you turned this around, and I started now with my disaccharide, and now the arrow is going to the left, and I'm going to break this disaccharide down into its individual glucose plus glucose, into its two monosaccharides. To make this happen, this water molecule over here actually literally has to be broken, lysed. And the OH from one side of that water is going to be put over here, and the H from the water will be chemically put here. Okay? So there's actually water being broken, hydrolysis, to get these two individual molecules back into their monosaccharide form. Now, last time I described a different word. I said that some chemical reactions were synthesis and some chemical reactions were decomposition. How would you therefore describe a condensation reaction? Yeah, it's another way of saying, or kind of similar to a synthesis reaction, right? You're making something. And a hydrolysis reaction, therefore, would be considered a decomposition, right? A decomp reaction. You're breaking things down, aren't you? Okay. Now, what was the other word that I used also when describing synthesis and decomposition reactions? I said that when we're building things up, the other word we can use for that is anabolic. And when things were being broken down, we could use the word catabolic. Okay. So I want you to have all three of those terms grouped together in your head. Right? Synthesis, anabolic, and in this case, condensation. Versus hydrolysis, decomposition, and catabolic. Have those categorized in your head. So I told you that sugars are the primary way that our body gets energy. Now, I'm not going to go through a deep lecture or two over cellular respiration. Who can tell me what cellular respiration is? Cellular respiration, the idea that we consume glucose and sugar, and that we're also breathing oxygen, and out of that, what does our body do? Breaks down the sugars until we get out of it what? Every day we blow off CO2, and we urinate, we get rid of water, and we also get energy out of that situation. There's a whole bunch of chemical reactions that allow that to occur. And they're listed here, and I'm not going to go into this. Another time and place. If you take microbiology, you will learn all that you'd ever want to know about glycolysis and the citric acid cycle. But I want you to look at this word, glycolysis, and break it down for me. Glycolysis, the breakdown of sugar. So glycolysis is the breakdown of sugar, and you'll be hearing about it in passing, but I'm not going to spend a lecture or two describing all of this to you. Just know that the sugars are the primary way by which our body gets its energy. Now this is what I do want you to memorize on the left. Okay? So this whole chart will be on the test on next Wednesday. No. Right. So this chart is a metabolic pathway chart. It is basically a collection of the vast majority of many of the chemical reactions occurring in your body. Now, what's the word for all of the chemical reactions in your body? Metabolism. So this would make a great shower curtain. Right? You might as well learn something while you're sitting there. Taking a shower, sitting in the room, and I know you can't see this, but right here in the very, very center, it says glucose right about there. Okay? And glucose then is going to be broken down. And as it goes down the very center of this, at the very, very bottom of this thing, it's going to say somewhere down here ATP. So what I'm saying to you is that all of your metabolism, all of the chemical reactions in your body, down the very center of it, glucose, right? The breakdown of glucose is critical. And as we break things down, energy is taken, and things like lipids, things like nucleic acids, things like other carbohydrates are made. Right? So our body takes in glucose, it breaks it down, it gets energy, right? ATP is produced, and then that ATP is used to create everything else that we need in our body. So this would be a chart of your metabolism or a chart of perhaps your biochemistry. Now, there may come a time where you'll get to learn all this. I can remember my first graduate course in biochemistry. We had to basically memorize this chart from top to bottom. And along the way, we learned every enzyme, every step. We also knew the structure of each of these molecules. And it was quite a course. A lot of memorization that semester. Okay. Now, what I want you to see here, though, is this equation down here on the bottom. And what does it tell us? Sugar plus water plus oxygen. Do you agree we're doing that every day? Not only is your body doing it, but every one of your cells is doing it. Every one of your cells is getting sugar. Every one of your cells is getting some water. Every one of cells in your body is getting in some oxygen. Okay? And then, what is your body doing with that? Creating waste products. It's creating CO2 that you're blowing off, water that you're urinating, and you're getting energy from it. This is the basic idea of cellular respiration. It's not hard. What do you do every day? Eat, breathe, drink. And every day, what do you do in response to it? Blow off CO2 and urinate, and as a result, you get energy out of the whole deal. Don't make it hard. If you take a general bio course, you'll break that down into greater detail. What's pretty much the opposite of this? Not exactly, but almost the opposite of this whole cellular respiration idea is photosynthesis, right? What plants are doing for you and me. So what are plants doing? What are plants doing? Plants are pretty much what? They're taking in water that we give them. They're consuming carbon dioxide from our breath, and they're getting energy, but their energy is coming from the sun, and they're creating, for us, oxygen and sugar. Right? We consume the plants, and we get sugar out of that. So it's pretty much the exact opposite. I shouldn't say exact, but it's largely the opposite reaction to cellular respiration. And that's all I'm going to ask you to know regarding carbohydrates. This table right here, I'm just going to have you just cross it off. I'm not going to worry about this table at all. Okay? On the bottom, it tells you that for every glucose molecule that your body takes in, we can make somewhere between 36 and 38 ATPs. Okay? So every glucose molecule that you consume in your body, you can make somewhere around 36 or so ATPs. And remember, ATP is the molecule of energy. Okay. But don't worry about breaking it down into the table. That's it for carbs. Any questions over carbs? Monosaccharides, disaccharides, polysaccharides, a couple of examples of them, why we care, and pretty much it. Okay, major energy molecule. Let's move on to lipids. Lipids, you might know them better as fats, but fats would be just one of the categories within the larger category called lipids. So lipids, you know them as fats, but also this includes oils. Now, the key property of lipids is that they are insoluble in water. That's going to be true of all lipids. They're all going to be insoluble in water, which means that they can't dissolve in water or they can't mix with water. And we know this to be true, right? Oil and vinegar can't mix. Oil and water can't mix. The water is, what's water? We know water is polar, a key word we keep coming back to. Water is polar. That means that lipids must be what? Non-polar. We'll see that word again. But here I'm calling those lipids insoluble. But another word you can put right there next to it is non-polar. Now we're going to go through a few examples of lipids. And the first one will be triglycerides. When you go to the doctor, they're going to check your blood levels, and they're going to check your cholesterol levels, but they're also going to check your triglycerides. Triglycerides are the most common type of lipid in your body, and what they are composed of chemically are 3 fatty acid chains, and I'll show those to you in a moment, and a glycerol backbone. So just know this, that triglycerides, 3 of something, right? Glycerol, there's 3 of these fatty acids attached to this glycerol, so triglycerol. Now there's a lot of energy here. Why don't you want a lot of triglycerides in your blood? What does it tell the doctor if you have a lot of triglycerides in your blood? What does it tell us about your diet if you have a lot of triglycerides in your blood? You've got a lot of fat, right, in your bloodstream. And in fact, you've got so much fat in your bloodstream, so much fat in your diet perhaps, that some extra of it is appearing in your blood. Now, fats are a great molecule for energy, right? You can eat just fats all day and do just well. Ask Eskimos, right? They eat blubber all day and they do just fine. And some of you may have in the past done an Atkins diet, right, or a low-carb diet. And what do those people, has anyone done a low-carb diet? Okay, what did you discover on that low-carb diet? What could you eat all day? Protein and fats. You could not consume carbs. And so you could eat, in fact, you didn't have to have that much protein. You could just sit there and eat butter all day. Blue cheese dressing, cheese, and butter all day, and you're going to get a lot of energy because fats give you a lot of energy in your everyday diet. Can you eat just fats and get by? Yeah. Your body, through that metabolic chart I showed you, has a way of converting fats into energy. You can also convert protein into energy and not consume any carbohydrates yourself. Okay. So lots and lots of energy coming from lipids. Now, this is a cartoon diagram of a lipid, of a triglyceride. Right? 3 parts. Let me break this down for you. So we see 3 of these molecules. 1, 2, and 3. And I want you to look at this molecule. What does that look exactly like that we saw last lecture? just the part that's in the box, looks exactly like, or really similar to, hexane. Go back. Where was hexane mentioned? Hexane was mentioned back when we were describing covalent bonds, and hexane was the example for non-polar covalent bonds. What page is that on?
20.
Page 20? Okay, slide
Slide 263.
263. Okay? So you see it talked about covalent bonds, and I gave you the example of hexane. Right? What is hexane? It was 6 carbons and a bunch of hydrogens running around it, right? And what was the characteristic of that molecule? It was non-polar. Now, what does that mean to you in everyday language? If it's non-polar, it means that it doesn't have any polarity, which means that every electron is being shared equally. There's no negativeness, no positiveness. There's nothing yanking up at the electrons. And every molecule in this every atom in this molecule is sharing the electrons equally, right? So when you look at a triglyceride and you see this fatty acid chain, of which there are 3, oh, you say that looks a lot like hexane. And I know that hexane is non-polar. Now, what's another word for non-polar? If it's non-polar, it's also if it's not, okay, let me do it this way. Non-polar, is it going to dissolve in water or not dissolve in water? It's not going to dissolve in water. Okay. Only things that are polar will dissolve in water. Things that are non-polar will not dissolve in water. So what do I have here? I've got a molecule that has 3 non-polar fatty acid chains on the side. And those 3 fatty kind of molecules are attached to a backbone called glycerol. Okay? There's 3 carbons in it. 1, 2, 3. And that's it. Right? We're not going to get fancy here. You don't have to memorize these structures. How do you think this molecule is put together? How is it knit together? Well, you're going to have 3 triglycerides attached to 3 fatty acids attached to 1 glycerol. And look what happens when we go from left to right. What kind of reaction is that going to be? I'm going to link them all together, right? I'm going to link all of this together with chemical bonds. So this is a synthesis reaction. We could call this an anabolic reaction. And we also could use the word condensation, because look what is going on in this reaction. In order to make this happen, I get 3 waters out of it. It's the same idea, right? We could take 2 monosaccharides and combine them into disaccharide and get a water. Now we're going to take 3 fatty acids, stick them onto a glycerol, and as a result, create 3 waters, right? 3 waters. So it's a condensation reaction as well. Now, what I haven't described is what's going to allow this to happen really, really quickly, is that there will be an enzyme, right? There's definitely going to be an enzyme involved. We're not naming that enzyme, but there definitely will be a biological catalyst that will speed this reaction up in your body. So that's a triglyceride. That's one example of a lipid, the most common lipid in your body. The other thing you'll hear about are saturated versus unsaturated fats. We hear that conversation every day, right? Look at your food labels. There are saturated fats and the unsaturated fats. Why do we care? Tell me why we care from a nutritional standpoint, and then I'll show you on here why it matters or what's going on with the chemistry. What is it about saturated fats? If I asked a doctor, even a dietitian, 25 years ago, are fats bad? The answer would have been all fats are bad. Old food pyramids showed fat at the very, very top of the pyramid to be used sparingly or none at all. Now what's on our new food pyramids? A whole sliver of the food pyramid is now fats, some of which are known to be healthy. What kind of fats are not healthy, or at least that's what we're thinking right now? What kind of fats do we want to avoid? Saturated fats. Now tell me, where does one find saturated fats? In potato chips, yeah. Maybe. But where do we find saturated fats in large amounts?
Oil.
What kind of oils?
No,
Olive oil is good for you,
vegetable oil.
right? Vegetable oils, many of them are not bad at all. So let me describe it to you this way. Saturated fats usually stay as a solid at room temperature. Crisco, bacon. Yeah, bacon grease, right? Bacon grease, Crisco, some of the margarines, the butters, right? Those things that stay solid at room temperature have a higher saturated fat. Unfortunately, M&Ms, any kind of chocolate bar. What makes that chocolate layer on the outside not melt in your hands? A high concentration of saturated fats, right? It doesn't melt at room temperature. So any kind of chocolate bar is going to have, even healthy, right, sports bars that have a chocolate on the outside layer are going to have a fair amount of saturated fat in them. That's what keeps that outer layer from melting, right? It stays solid at room temperature. Now, unsaturated fats are ones which are tending to be liquid at room temperature. So they're going to be oils. They're going to roll over and roll over. The molecules are going to roll over each other. So this would be your olive oils and some of your vegetable oils. Some of them aren't so bad for you. Some of them have a higher concentration of saturated fats. And then there are ones that are being modified chemically, and some of those we are now finding are not the best thing for our bodies. So people got away from butter for a long time. Why? Because it was full of saturated fats. So we took vegetable oil, which we knew was healthier, but to make it stay solid at room temperature, we hydrogenated it. In other words, we monkeyed with vegetable oil in the lab to create a butter-like substance called margarine that stayed solid at room temperature. And we thought it was healthier for us because we were using vegetable oil. And now we know that those hydrogenated oils might be even worse than going for butter, right? Just stay with the natural butter. It might be better off for you than the hydrogenated oils, okay? And then there are some oils like coconut oil that, even though it's an oil, palm oil and some of those oils that you'll see in food labels, even though they are oils, they are really full of saturated fat. So it's not an all or none rule. Some oils are good for you, some oils are not good for you. And there was another report out just last week or two about how butter, in the end, might be better than all this other crap that we're eating that are these modified food substances. So going back to nature might be make more sense. And you know, we're not surprised, perhaps, as we start looking at some of these reports. Now, chemically, what do we have here? Look at this molecule. Okay, what do you see? A long chain of carbon, C, 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 C. The line between them is a chemical bond, right? Electrons are being shared. A bunch of carbons, long chain of carbons, and what do we see around every single carbon? Hydrogens, right? The hydrogens are saturated. There is no more room for more hydrogens on this molecule. So this is a cartoon representation of a saturated fat. It is saturated with hydrogens. Now, at room temperature, this fat would tend to stay solid. It doesn't roll over like an oil. It doesn't melt easily, okay, or as easily. Look below it. What do you see? I see a long chain of carbons, but I see an occasional double bond. Now, I'm not getting into the chemistry of all this, but you see the equal sign, right? Some of these have an equal sign. Now, where there's an equal sign, that means that it's two pairs of electrons. And what do you also see missing? Wherever there's an equal sign, you also see some H's missing. These are not saturated. They are unsaturated fats. So notice there's a gap, right? There's no hydrogen here, no hydrogen here, no hydrogen here. Now the lack of that hydrogen allows this molecule to pour, to roll. So this kind of molecule is going to stay liquid at room temperature, right? Oils, right? And they tend to roll over. The molecules bend and are more pliable. The saturated fats tend to stay solid, and that's what we think clogs up our arteries, right? So the thought was, okay, saturated fats are bad for us, and it's true. We don't want too many saturated fats in our diet. But saturated fats are the ones that we thought clogged our arteries, whereas the unsaturated or the polyunsaturated fats were the ones that were healthier for us and did not clog up the arteries. So now you see this idea of saturated versus unsaturated. Just know that saturated, they are saturated with hydrogens, right? Whereas unsaturated are not, and they are the healthier of the oils. Now, what did I just say a moment ago? What did manufacturers do? They took unsaturated vegetable oils and they synthetically what? Hydrogenated vegetable oil. And by taking this molecule on the bottom and artificially saturating it, what happened to this molecule on your countertop or in your fridge? It no longer was an oil, now it was a solid. Yeah. Okay. So that's what we did in the chemistry lab to make what we thought was a healthier oil behave like butter, so we could spread it on toast. Another common example of a lipid, phospholipids. These are a group of very important molecules that are in your cell membranes. So your cell membranes, your plasma membranes of your cell are largely made up of phospholipids. They're lipids, right? And the lipid part tells me that they are what? Non-polar. But the phosphate portion on these molecules gives them a polarity. So what I'm telling you now is that one end of these molecules is nonpolar and one end of these molecules is polar. One end of these molecules can interact with water. And we're going to use the word hydrophilic for that. So a molecule that's hydrophilic, hydro, water, philic, a love of or an appreciation for or an affinity toward. Right? So hydrophilic is water loving. Things that are water loving are going to be polar. Okay? Whereas, so I'm telling you that one end of the phospholipid is polar, so it likes water. The other end of the phospholipid is non-polar. Another way of saying that is that it is hydrophobic. It has a fear of, moves away from water. Hydrophobic. What's the other word we want to use here? I'm describing polar and hydrophobic. The other word, well, polar, right? And hydrophobic, hydrophilic. No, it's okay. We're okay. We got polar and hydrophilic together, non-polar and hydrophobic together. That's good for now. Let me show you a picture of a phospholipid. Cartoon of it. So this long thing right here is called a this is your fatty acid. So that's a bunch of carbons and hydrogens, isn't it? Okay, it's a bunch of carbons and hydrogens all linked together. And what do we know about that long chain of carbons and hydrogens? It is what? Yeah, nonpolar, or another way of thinking about it, hydrophobic. Right, that's like the fatty acid chain. Now, in this phospholipid molecule, though, up here, this region is the phospho group, the phosphate group. Now, the phosphate group is polar. That is, it is hydrophilic. Okay? Polar or hydrophilic. Now, in other cartoons, rather than showing what's on the left, they'll show you a more simplified molecule like what's on the right. They'll show you this little blue circle. That's the phosphate group. And then they'll show you two long long chains coming off from it. Those are the fatty acid chains. Those are the lipid-like extensions. So that's a cartoon of a phospholipid. Now another example of lipids, the last example for now, are steroids. You've heard of steroids. We hear about them in the news on ESPN for people who are taking anabolic steroids, right? Molecules to build up muscle. But you also have in your body things like cholesterol, estrogen, progesterone, testosterone. All of the sex hormones are steroids themselves. They're lipids. They're fat-like molecules. They're all chemically related to this. This is a cartoon of cholesterol. I don't need you to recognize it, but it looks very different than anything we've seen so far. And there are 4 of these chains, of these rings-like structures. So this is cholesterol. Now cholesterol gets a really bad rap at the doctor's office, right? We want to keep our cholesterol levels low because we think that cholesterol is an indicator, high cholesterol levels is an indicator of potential heart disease, right? Higher cholesterol levels in the blood might suggest that with time you'll have heart disease. Well, the data on that's not that clean. It's really not that clean at all. There have been studies for years showing that in some populations, people with high cholesterol do not go on to develop heart disease. We don't have this completely figured out, but the pharmaceutical companies love this story because the moment you have a higher cholesterol level, they're putting you on some sort of medication for the rest of your life to help keep your cholesterol levels down. And there's some truth to this, but like all things, it's not a straight line. It's not more cholesterol means more heart disease necessarily. Now, these molecules, again, they're all lipids, and they can go straight through plasma membranes. Okay, more about this later, but steroids can go right through your skin and right through your membranes. This is why right now you can't turn on the TV without hearing about low T levels, right? Some guys got low testosterone levels. And the medication for this, because guys are wimps and don't want injections, guys are spreading on this stuff under their arm, right? It's a lipid. It's a cholesterol-like molecule, and it's absorbed straight through the skin and into the guy's bloodstream. And then at the end of the little commercial, what does it say? Women, young children, women who think should be pregnant, should not come in direct contact with this stuff, or snuggle up with the guy who just put it under his armpit, because this testosterone can go right into your body as well. Now, so you would not want to have a woman get a lot of this testosterone, especially a pregnant woman, because if the fetus is a little girl, then that testosterone can start influencing the little girl's genitalia and her sexuality and her sex organs, and it can be a real bad situation. We'll talk more about that later on, but my point now is this: steroids are lipids, and lipids can go straight through skin, and lipids can go straight through plasma membranes. Why? Because lipids are non-polar, and we'll see later on how the cell membrane also allows non-polar molecules to go straight through them. Okay, so if I said lipids, what's going to come to your mind? Check off some ideas of lipids. Fats and oils, saturated or unsaturated, triglycerides, phospholipids, and steroids. They're all part of the lipid family. Okay. That's all we'll deal with that. Any questions on carbs or lipids? Carbs or lipids? Okay, the next of our 3, proteins. So proteins are built up. We said that carbohydrates are made up of monosaccharides and disaccharides. We said that fatty acids, as an example, are made up of, or triglycerides are made up of fatty acids and glycerol. Proteins are another group. They are made up of amino acids. Now, it turns out that in your metabolism, there are 20 different amino acids. You've seen some of them on your vitamins. Some amino acids are considered essential amino acids. Why? Because your body needs them. They're essential for life, and your body can't make them. Other amino acids your body can produce, and so you don't find them on your nutrition labels or on your vitamins as much. But the amino acids that you need, because your body can't make them, are oftentimes in higher level vitamins, right? Good vitamins will have some of these essential amino acids in them. Now, there's 20 different types of amino acids, and let's just look at this name, amino acids. Well, it turns out that amino acids get their name because each of them, all of them, have in common an amine group, NH2, and an acid group, it's called a carboxylic acid group, COOH. Okay. So this is why they get their name. They have an amino group, they have an acid group, they're the amino acids. Now, we're going to talk about proteins and the different things in your body that are made up of proteins. Don't forget that enzymes also are proteins. Right? Enzymes were those biological catalysts that ended in usually in ASE and were able to speed up chemical reactions. So what do proteins do for you? Well, all the structure of your body. All the structure of your body is made up of proteins. So collagen, you'll hear about collagen. It's a very important protein. It gives you much of your structure. Your bones are largely made up of collagen. The enzymes, I already mentioned the enzymes, those biological catalysts that are going to allow chemical reactions to occur or to not occur. Protection, your hair and your fingernails are made of this protein called keratin. It's a protective kind of protein. Your muscles can't change shape. They can't contract without proteins called actin and myosin. And then lastly, as an example, your blood contains it within it hemoglobin. And hemoglobin is going to transport your oxygen and CO2. So we're seeing lots of examples, just really quick, examples of proteins. Not all, but many proteins end in IN. So you see hemoglobin, myosin, actin, keratin. Some of them break the rules, but most proteins are going to end in IN. Remember, sugar is ended in ose. Most proteins will end in IN. I mentioned enzymes. I mentioned to you before that little curve. Remember that curve that I drew for you? It looks something like this. It said that an enzyme does what? An enzyme, here's the hump for the chemical reaction to occur. And with an enzyme, the hump is lower, isn't it? Okay? So that was kind of a picture idea of what an enzyme does as far as the energy. Now, what is an enzyme doing? It's allowing for chemical bonds to be created or changed more easily. That's really what they're doing. I like doing this graph from right to left at first. So what I'm showing you is in this picture, the enzyme is this blue protein here. Now this enzyme has a very unique shape. Notice it has a very unique shape to it, and we'll discuss this as like being a lock and key, right? Very unique shape. Now the products are here. I'm going to call these the reactants because what I want to do is go this direction with this equation, with this whole thing. So we've got two things here, the purple one and the orange one, and this enzyme is allowing them to come into close proximity. And they come into close proximity here, and then they create something. They are combined. Okay. So this enzyme can go either way. It can go from left to right or right to left. But I think most of us think about enzymes as building things. So on the right-hand side, these would be the reactants, and this would be the product. Okay? So we've got reactant A and B making something called C, right? A and B are combined to make C. That's one thing that enzymes do. Now, the other thing that enzymes can do is go the other way. So now what would we have? C would be considered what? If we're going from left to right, C would be considered the reactant, and the products would be A and B. It doesn't matter. Right? You can go either direction with this kind of image. Remember, that's what enzymes do, and enzymes are proteins. I wanted to remind you of that. That enzyme, just a moment ago, had that really funky lock and that key kind of shape to it, that certain groove on the edge. And so proteins do their job largely because of their shape. Or another way of saying it, the shape of the protein is critical to its function, right? And that brings us back to lecture 1, where I described anatomy and physiology, that shape and form, or shape and function, always go together. So again, proteins are made up of amino acids. There are 20 different amino acids. And I'm going to talk to you about primary versus secondary structure as we go through this. Primary structure is one amino acid linked to another amino acid linked to another amino acid, making a chain of amino acids. So the primary structure is a chain, right? It's a chain of amino acids. Secondary structure, as we're going to see in a moment, is caused by hydrogen bonds. Remember hydrogen bonds? I talked about water a little bit, and I said, you know what, in water, the oxygen from one water will be attracted to the hydrogen of another water molecule, right? So that would be a hydrogen bond right here. And we're going to see secondary structure is how these amino acids interact with each other, how they bend and twist when they're in close proximity with one another. That'll be the secondary structure. So let me show you a picture of this. Across the top, there's your primary structure of a protein. And what do we see? One amino acid in a chain after another one. Don't worry about the abbreviations right now, but each of those ALA, LU, SEER, GLU, GLU, HIS, those are different amino acids. So we're just seeing a bunch of amino acids in a linear chain. That's considered the primary structure. The secondary structure, again, caused by hydrogen bonds, oftentimes causes proteins to have different overall motifs or shapes, like a random coil or maybe a twisted helix like this, or maybe a sheet-like structure like this. Now, all of these amino acids interact one with another to create their tertiary structure. That is their third level. And this is the final level that we're going to discuss right now. Proteins get in their final structure. Every protein has a particular shape. In this cartoon, this is that green protein wrapped around on itself, and that would be the final shape of that protein. If I went back and I looked at go back and look at that enzyme, this would be the final, this would be the final tertiary shape of that enzyme. Right, so every enzyme has a particular shape, and if it doesn't have the right shape, it can't do its proper job in the body. So we're going to stop with tertiary, not go on to quaternary structure right now. We'll come back to that later in the course, but for now, just stop at tertiary, and you can just think that this tertiary structure is the final three-dimensional structure, right? So tertiary, this is the final three-dimensional structure of the protein. So you know that the shape of a protein is critical to its job. What happens if we change the shape of the protein? What if we unravel it or change its three-dimensional structure? And that is called denaturing. So if we denature a protein, that is, we break it apart, we no longer allow it to maintain its shape. This can be done rather easily by one of two ways. One, you can raise the temperature, or you can lower the pH, make it more acidic. So if I take a protein and I raise the temperature, it will begin to unravel. It will begin to loosen apart, and now I will lose my three-dimensional structure. Once I lose my three-dimensional structure and the temperature comes back down, proteins don't just poof, pop back together. So once destroyed, destroyed. Once you destroy this protein or denature it, it cannot renature very easily at all. So I can do this through either increasing the temperature or I can lower the pH. I can take a protein, put it into acid, and it will also become denatured, disrupt the shape. Now, this is what happens in your gut. As you're eating things, molecules, proteins are denatured in your gut because your gut has a very low pH. That's a good thing. I mean, it helps to break down food. It helps to break things down into their smaller pieces. But you wouldn't want your blood pH to drop, because now all the proteins going through your blood would begin to denature. Now look at this protein in this cartoon, and let's just make sense of this. So here's your squiggly three-dimensional protein, right? I'm just going to draw this all over again, but you got this three-dimensional protein. That's its final shape, that's its proper shape. And there are parts of this protein that interact with this molecule. So this is the enzyme down here, and this is its substrate, that is its reactants. And these three, the red, yellow, yellow amino acids, are the ones that fit into those little grooves on the substrate. So we have to have the red, yellow, yellow side by side for this protein and enzyme to work properly. If we denature the enzyme, though, look what happens on the right. Now we let go of the three-dimensional structure. And now the yellow amino acids are way down here, and the three of them are no longer together. Therefore, they cannot interact with this molecule. And basically, the enzyme has been denatured. It no longer has any purpose or function. So we don't want our proteins to become denatured. This is the problem with high fever for a long time. High fever in an individual, the body temperature is going up, and the enzymes are beginning to be denatured, and at some point it can cause permanent damage, or in a young child, permanent brain damage, if that temperature is elevated for too long of a time. That's proteins, right, in this little introductory thing. Let me stop right there and give us about 5 minutes just to clear our minds. We'll come back and we'll finish up with nucleic acids, and then that'll finish up the content for chapter 2. Okay, so what questions do we have so far on proteins, nucleic acids, or not nucleic acids, but carbohydrates, lipids, or proteins? Anything else? Anything that can come to mind right now?
Probably a stupid question, but when we're talking about joining things together and getting water, what form do they start out in? Like when we were talking about the monosaccharides, the glucose plus the glucose.
Right.
The disaccharide, and then it says you get an H2O out of that. So is that then become a liquid because of the water molecule? I mean,
No, okay,
how does
so that water molecule is simply made as a result. So the question was, where does all this thing start? So we've got a monosaccharide plus a monosaccharide undergoing a chemical reaction where they're formed together to make a disaccharide. And as a result of that, it's a condensation reaction and a water molecule is formed. You're just making water. You're just making a water molecule, and now you have, instead of two glucose molecules, or a glucose and a fructose, now you have a sucrose. Okay.
So it doesn't necessarily change the form it takes then?
It went from being glucose, now it went from being an individual. The water is the water.
Okay, so
Water
it's
is
not
just,
like it's, like a solution or something. It's just
no, that water is still just a free water. So every day, let me just put it this way. Every day, you eat and drink about 2,000 milliliters of water. You gain that water through your food and through your drink. But you actually are creating in your body water as well. You're creating water. And then you get rid of that water through your urinary system, through your kidneys. But you're actually creating water in your body. How? By these condensation reactions. So as you take a glucose and you put them together to make maltose, you've just created a molecule of water. And your body's doing this all the time. When it makes proteins, when it makes fatty acids, when it makes carbohydrates, it's creating water, right? And you're adding water to your overall metabolism. So you're not only are you consuming water, you are making water. Okay, so that's the first three of the four groups. Now let's go to our final group of macromolecules, and that is nucleic acids. Now the nucleic acids are built up of building blocks called nucleotides. Nucleotides. And you know the nucleotides. You've heard about them most of your life. You may not know they were called nucleotides, but this is the A, C, G, and T that we hear about. Right? So that's basically the nucleotides. Now, a nucleotide is a little bit more than that, but let me tell you what it is officially. A nucleotide is officially a base. Now, that base is called the A, G, C, or T. That's the sugar group. And that sugar group comes in two flavors. It can be a ribose or a deoxyribose. Notice it ends in OSE. It's a sugar. And then attached to it is a phosphate group. So let's take a look at this nucleotide. What we have is here's the base, this section here, chemically attached to a sugar, and then attached to a phosphate. Again, you're not going to be responsible for drawing this out or recognizing it, but you do need to know in your head that a nucleotide is a base plus a sugar plus a phosphate. Right? A base plus a sugar plus a phosphate makes up the nucleotide. Now, where are we going to see these nucleotides? In DNA and RNA. We'll get started there. DNA, you know, I know that you know, that DNA is a double helix. We hear about that all the time. DNA is a double helical molecule. We didn't know this before in the 50s, but for the last 50 years, 60 years now, we've known this to be a double helical molecule, which means basically over here on the side, what do we see? We see it looks like a winding staircase, don't we? And on one side of the staircase is linking over to the other side of the staircase. And in between, what do we see? The bases. What we see here is that, for example, G always is matched up with C, or T is always matched up with A. These are the nucleotides, and they're along this molecule. Now, DNA is, you could argue, the most important molecule in your cells because it is regulating all of your biochemistry. It's regulating everything about you. It's making up your genes, your chromosomes, all of that. So that's DNA, and we're going to discuss more of this when we get to chapter 3. But I also want to compare DNA with RNA. Now the D in, let me go back, the D in DNA is because of deoxyribose. So the sugar in DNA is deoxyribose. Whereas the sugar in RNA is going to be ribose. Okay? Can't write today. Now RNA, there's a whole bunch of different types of RNA. But RNA, all of them are relaying information in some way. They're all building blocks or relay molecules. And what we'll see is that we're going to compare a little bit about DNA and RNA. RNA is going to be a single-stranded molecule, whereas DNA is going to be double-stranded. So that's one of the big things, right? DNA is double-stranded, RNA is single-stranded. Here's a picture side by side, comparing a few more. So on the left-hand side, RNA, on the right-hand side, DNA. We can see that the DNA is double-stranded, right? So the DNA is double-stranded, whereas the RNA is single-stranded. The backbone is the same. In other words, if this were a winding staircase, I see sugar, phosphate, sugar, phosphate, sugar, phosphate. So the backbone is the same, no matter if I'm talking about DNA or RNA. Another difference, though, is that in DNA, the bases are A, C, G, and T. Whereas over in RNA, there won't be any T. Instead, there will be U. So there will be uracil. If you see uracil, if you see a letter U anywhere on a molecule, you know that that molecule is an RNA molecule. If instead you see T's, you know that that molecule is a DNA molecule. And DNA is found within the nucleus of the cell, and DNA is a big part of what makes the chromosomes. So those who are in lab this afternoon and those who will be in lab tonight or on Wednesday, you'll be looking at cells undergoing mitosis, and you will actually be able to see the chromosomes in the cell as the cell is dividing. And this is a cartoon of that chromosome. So up here at the very top of this diagram, that's my double-stranded DNA. Right? I can see the double-stranded DNA. And what will happen is that that DNA will wrap very, very tightly around little proteins. And these proteins are called the histones. Well, I'm going to tell you right now, if I go back a slide, that this phosphate group, going back 3 slides, 4 slides, see this phosphate group? This phosphate group has a negative charge on it. See those negatives? So a phosphate group, by its very chemical nature, is negatively charged. So DNA is, by its nature, negatively charged. Now if I go over then and look at this picture of the chromosome, and I'm pointing out to you that the DNA is wrapping very tightly around these little proteins. What must these little proteins, what charge must be associated with them? Yeah. So those little proteins must have a positive charge on them, acts like little magnets. And what happens is that the DNA wraps around these positively charged proteins and creates a more dense structure. And then those little wrapped around DNA molecules will then form what looks like a telephone cord. Right? A very densely wound telephone cord. Now, most of you in this room have never seen a telephone cord. Okay? But if you'll go to Washington, you can go to the Smithsonian, and there you can go over and you can see a telephone cord, right? We used to be tethered to a telephone cord, and it would get wrapped up, and it kind of looks a lot like that. Now, under the microscope, what you're seeing is this letter X. When you look under the microscope, you're going to see these X-shaped sort of like chromosomes. But what I want you to know is that what you're seeing is a very densely packed, right, DNA wrapped around proteins, and that collectively makes up the chromosome. So DNA, a very, very important part of our cells. I've got just a couple slides to show you for the end of this material. I've mentioned ATP a couple of times. In lab, I'll mention ATP this week. ATP is the major energy molecule for the cell. And in biology, when you hear the word energy, typically ATP is the currency or the molecule of that energy reaction. Now, I think it's really cool when you start looking at biochemistry and how the body uses different molecules to do its jobs. ATP, this molecule we're talking about for energy, if you broke it down into pieces and parts, it's an adenine. That's the same thing that's in DNA and RNA, the A. And it's a ribose, which is the sugar group found in RNA. And it's not one, but it's 3 phosphate groups. That's the tri. Okay, so collectively ATP means adenosine triphosphate. It's an adenosine, it's an adenine with a ribose, and 3 phosphate groups. Now, what did I tell you about chemical reactions? When chemical bonds are formed and put together, it usually requires energy. When chemical bonds are broken and things are catabolized or broken down, typically energy is released. Well, that's exactly what happens in ATP. It just happens to be that this particular bond right here, this little squiggle, is what we consider a high energy bond. And when ATP breaks that bond, when that phosphate bond is broken, it releases an abnormally large amount of energy. It's just an abnormal amount of energy. But it's stored, right, in this ATP, and then when the cell needs energy, it breaks this bond right here at the red arrow, and the cell uses that released energy to do some work, right, to create some work. So that's the molecule of ATP. And then lastly, I want you thinking about this, anabolic versus catabolic. So what was catabolic? Breaking down. And when you break things down, right, when ATP is broken down catabolically, we get out of it energy. Instead, we also have to make ATP, right? We have to make this ATP. And when our body makes ATP, then the body is having to do an anabolic reaction, isn't it? To make the ATP, to build it up. Again, where do we get that? Where do we get the energy to make ATP? From our diet, right? From our diet. So we're consuming food so that our body can build up ATP, and then that ATP is used throughout our body to create other molecules. Just the last slide is a reminder slide of the building blocks. So when I think about polysaccharides, sugars, starches, glycogen, where do they come from? They are longer units of more simple monosaccharides. So monosaccharides combine to make polysaccharides, glucose, fructose as examples of the monosaccharides. If I'm discussing amino acids are made up of proteins, right? Proteins are made up of amino acids. So amino acids are the building blocks of proteins. Right, so proteins are made up of the amino acids. And then finally, how are we going to make nucleic acids? We're going to take individual nucleotides, right, and build them together and create the DNA and the RNA. So it's important you know these monomers and these polymers, these individual units that build to make the larger units. And that does bring us to the end of the material for the first exam and to the end of the material for, for the first exam. So that's chapters 1, chapter 2, and the material from the first lab. Anything at all that you're wondering about? So we'll get started into chapter 3 right now, but is there anything at all from chapter 2? Question?
There's a question on the quiz.
There's a question on the
We go to AMP
mastering, okay.
Mm-hmm. But there's a question, the structures of RNA differs from DNA and that. And is this all we're going to talk about, the RNA and DNA?
At this point.
Okay, because none of these answers make sense to me. Like it talks about,
Yeah.
when I tried to look through the book, I couldn't find anything that would make sense.
Look at the second choice. The RNA is made up of the sugar ribose, whereas DNA is made up of the sugar deoxyribose. Okay. Now we will dive a little bit deeper into DNA and RNA into chapter 3. And that question will make more sense then, it really will. But for now, you can still answer that, but you got to do a little bit more work on that question. But as far as what I'm going to quiz you on, will be directly from what we've discussed here. So, okay? So know that. Know that I'm only going to quiz you on the things directly that I've described in the PowerPoints or in my conversation with you on this first exam. Now on Wednesday, you can still come on Wednesday and ask questions about things on the exam. So I'm hoping that between now and Wednesday, if you haven't already, that you go into mastering, look at homework 1 and 2, at least play with them, interact with them, see where your questions are, where your concerns are. Look at your book. If you haven't had a chance to start looking, I think you're really going to enjoy the book as you take a look at it, and then come back on Wednesday with any questions that you might have related to exam number 1. Now, the reason we're having the exam so late, why it's a whole week from now, is partly because of Memorial Day, and partly because I wanted to make sure that everybody had a chance to go through Lab 2 this week about ionic and covalent bonds and solubility and pH before we have our exam. So the exam would have been next Monday, right? But because of the holiday, it got pushed even further. So don't get too, lax. We got 10 days away, but go ahead and just study this material and be really confident with it. Now, that also means, though, between now and next Wednesday, I'm going to start discussing some new material with you right now. So the material from this point on is Chapter 3, and this material is different. It's going to be on the second exam, so it'll be a while before you'll be tested on this, but I think you'll also find that it's going to help you with your understanding of Chapter 2. Now, I don't think your PowerPoints say Chapter 3, right? It just kind of has this blob on the picture, and this is the beginning of Chapter 3 in your PowerPoint lecture notes. Let's go ahead and quiz, though, right now on this. This is a good thing for the first exam. Review what section, what plane was cut through to see image A. Everyone sees sagittal. You could have said mid-sagittal, but sagittal is definitely correct. Remember, how do we spell sagittal? 1 G and 2 T's. Letter B, or B. Frontal, also called coronal. Coronal and frontal are exactly the same thing. You need to know them both. That's what was used to create B. And then in letter C, what do we have there? Transverse, also called cross-section, right? Either one, same thing. Cross-section or transverse. Good. So cells. I think they're super cool. My undergraduate degree was cell science. My master's degree was microbiology and cell science, so I'm kind of geeked about cells. But I promise you I won't over-geek and cause you to learn more than what you need to when it comes to the cell. But we discussed in the hierarchy from atom to atom that cells are the building blocks of us. They are the fundamental living structure within us. So we have within our body trillions and trillions of living cells which have created this whole thing that we refer to as the organism. About 75 trillion cells in your body. About 75 trillion cells. And there are actually, though, about 200, I put over 200, some books will say as many as 250 different types of cells. So you don't just have, muscle cells and kidney cells and liver cells. There's actually over 200, maybe as many as 250 different types of cells in the body that each have their own job, some of which we don't still understand what they do. We're still learning about cell biology. You would think by now we would have all the bones and all the anatomy figured out. And that's true, we have most of the structure figured out, but we don't have all the physiology structure figured out at all and what cells are doing. So there's still a lot of research going on about the human body. Now, what do we know about cells? They're the most fundamental living thing. So as we described in the first lecture, if a cell is living, therefore it must have all the characteristics of life, and that would include obtaining nutrients and using it. That's a fancy word of saying that a cell has a metabolism, right? It takes in nutrients and uses it. It gets rid of their waste products, also a part of that metabolism. It has a shape and integrity, meaning that it's made up of smaller units, right? Organelles and structural things. And it can reproduce and divide through cell division, through mitosis. So it has all those basic requirements of life. Now, size-wise, scale-wise, can you see an individual cell? The only cell in the human body that is possible to be seen with the naked eye is the egg. Okay, the human egg is about as small as a little pin drop as you can imagine, but it would be visible with the naked eye. So I'm looking here, and I see it says the human oocyte, the human egg, and it's right at that border between what you can see with the unaided eye and with a microscope. In fact, you'd be much better off to see it with a microscope, but you'd be able to appreciate it. If someone said that little thing on the paper was an egg, you could say, okay, I can see that little dot. Other than that, you're not going to see a cell, right? Cells are going to be microscopic. So looking at this figure, what can we see with the unaided eye? Well, clearly we can see entire muscle, right? There are some nerves in your body which are large that you can see, but you can't see an individual neuron, the cells within the nerve. We can see an ostrich egg or a chicken egg. Those are much, much larger. In order to see most of this, we have to use a microscope. So this semester, as you know, we'll be using the microscopes. We'll be using the microscopes in order to see the cells, and that will include red blood cells. And even when you take microbiology, if you take that course, you'll use the microscope a lot to see bacteria. And you can use a light microscope to even see large organelles like mitochondria. You can't even, but to see below that, I should say, you're going to have to use a different kind of a microscope, an electron microscope. It's not something we hang around in our back pocket, but in large labs they have electron microscopes, and with electron microscopes now you can see viruses, you can see ribosomes, you can see even large proteins and lipids like what are found in the membrane, and even some small molecules that have been seen. What we're not yet, and I may be off a little bit on this, but my understanding of it is we still don't have the ability to see individual atoms with an electron microscope. I mean, we know they're there, sort of like the wind. We have evidence of it, but as far as actually seeing an individual atom, I don't know that we're there yet. But we're getting the sense of how small, right? We can see things with an electron microscope. So most cells are going to be between 1 micrometer and 140 micrometers in diameter. The 140 would be the human egg, and it is, as I mentioned, big enough to see with the naked eye. Again, we typically have to use a light microscope, and then if we want to look at subcellular structures, we're going to have to use an electron microscope. What do cells do for us, for our body? Well, they're part of our tissues, right? Cells make tissues, and our epithelial tissues and epithelial cells are covering our body, our skin, and they're lining our internal structures. For that, I want you thinking serous membranes, right? The serous membranes, the pleura, the pericardium, the peritoneum. Those are membranes or layers that cover and coat and line our organs and line the inside of our tubes. Storage. Adipocytes, fat cells, adipose. Not only are we storing energy, or fat and energy. Movement, right? Without cells, I told you a moment ago that proteins were necessary for movement, but without cells, too, we wouldn't be able to move our body. So we have to have muscle cells to contract to allow our body to move. Ligaments, tendons, things that connect bone to bone or bone to muscle, tendons, are the function of cells. We have a lot of cells involved with our immune system. White blood cells are attacking foreign objects, macrophages, other cells that are part of our defense system. Nerve cells, neurons are communicating throughout our body. Egg and sperm are necessary for reproduction. So we've got lots and lots of different kinds of cells within our body. Now here is a prototypic cell. In pre-lab coming in, next week's pre-lab, you'll be labeling the parts of a cell. So you're going to get to practice this again. What's that? This week, and you'll see some more of it next week as well. Yep. So you're labeling part of the cell, and that'll help you, right, as you move forward. Now, this cell would never exist in your body. This is a prototypic example, and there are some things found on this cell that would never coexist with other things found on this cell. Let me just walk you through this, major ideas on the organelles. So we'll start in the very, very center of not every cell, but many cells, and that is there's going to be a nucleus. Now, the nucleus is where the DNA is found within the cell, It's where the chromosomes will be. And typically, there'll be a much darker staining area in the center of the nucleus called the nucleolus, or you'll hear nucleolus as well. What we'll be focusing on in this chapter is that you'll be able to recognize each of the organelles, parts of the cell, and then you'll also need to know what each part of the cell is doing. So what does each organelle do for the cell? If you think about it too, from our atom to atom continuum, chapter 2 was atoms, molecules, and macromolecules. And now this chapter we get into the organelles in the cells. So we have the nucleus and the nucleolus. Outside of that, there's going to be a whole series of membranes called the endoplasmic reticulum. Endo means within. What is that same as? No, the nuclear envelope is the membrane around the nucleus, right? The envelope around the nucleus. Outside of that, though, even more, is the endoplasmic reticulum. Now, endo means within. Plasma, it's just another set of plasma membranes. And reticulum is a word we're going to be seeing all over the course. And reticulum means a network. So it's a network of membranes found within the cell, right? Endoplasmic reticulum. And endoplasmic reticulum is found all around the nucleus. Now, I don't want you to get stuck thinking that this is, don't forget, this is a three-dimensional cell. And the nucleus is spherical. And the endoplasmic reticulum, likewise, would be going all around, right, all dimensions of this sphere. But whenever we have a picture, we got to kind of cut it some way. So we have the endoplasmic reticulum. Some of the endoplasmic reticulum will have ribosomes attached to it. You'll see these little tiny dark dots all over the place. So there's ribosomes on the rough endoplasmic reticulum. And then, as we'll see, there are going to be areas of endoplasmic reticulum where there aren't any ribosomes, and that is instead called the smooth endoplasmic reticulum. And it has a different purpose. We'll describe that. Right now I'm just kind of labeling the parts. Also inside the cell, there'll be another set of membranes. It always reminds me of that old-fashioned Christmas ribbon candy. Called the Golgi apparatus, or the Golgi body. You'll see it, or just called the Golgi. Named after a person, Dr. Golgi. And so that one's always capitalized. And the Golgi is another set of membranes, like I said, and we'll see what happens there in a few minutes. Now, around the outside of the cell, plasma membrane, also called the cell membrane, all around it. Again, this is another organelle. An organelle is any functional part of a cell. So any of these things can be considered an organelle. Also going back inside, though, I see these bean-shaped organelles. Those are mitochondria. Okay, they're all over the place. And the mitochondria are going to be the energy-producing organelles. They're going to be making the ATP for the cell. There are these little guys. They look like some sort of connectits or some sort of Lego blocks. Those are the centrioles. Okay. And those who were in lab today talked about mitosis. And we talked about the mitotic spindles reaching out. They start at the centrioles. Okay, so they're important for mitosis. What else do we see in here? All the way through the cell, I see some strings, what look like, structure. And that's exactly what they are. All the way through there, you'll see what's called the cytoskeleton. Not only does your body have a skeleton that gives you structure, so too every cell has an internal structure or skeleton, cytoskeleton. Okay, and all those little proteins in there are hard to see, but they're giving the cell shape. There are going to be a series of vesicles. Now, some of these vesicles are called lysosomes. There are other names for them. We'll look at a couple other examples in a few minutes. But there are peroxisomes and there are lysosomes, and they are going to collectively carry different things around in the cell. Everything inside the cell can be referred to as the cytoplasm. Okay, so the liquidy portion and everything inside the cell is the cytoplasm. And then on the outside of the cell, we see a couple of things here. I'm seeing a few really long processes. These would be